Welcome to the fascinating world of early brain development. Today, we are going to explore a magical and crucial process called neurulation. This is the very first step your body takes to construct your entire nervous system. We will build our understanding from the ground up, looking at both the biology and the physics behind how our brains begin to form. Now that we know what topic we are exploring, let us start with a simple analogy. Imagine you are lying in a sleeping bag that is completely unzipped and laid out flat on the floor. To get warm, you grab the edges and pull the zipper all the way up, turning that flat blanket into a cozy three-dimensional tube. In the very early stages of human life, your body does something incredibly similar. The embryo starts with a flat sheet of cells called the neural plate, which is completely open and exposed. To create the complex central nervous system, this flat sheet must fold up and zip itself closed. Building on that sleeping bag analogy, let us look at the formal biological terms for this process. Neurulation begins when a specific layer of embryonic tissue, known as the ectoderm, thickens to create the neural plate. As development continues, the lateral edges of this plate, which we call the neural folds, gradually rise upward. These folds eventually meet in the midline of the embryo and fuse together. Once this fusion is complete, the resulting hollow structure is called the neural tube. This delicate folding and fusing is a highly coordinated event that must happen perfectly to ensure healthy development. Having seen how the neural tube forms, we must understand when and why this happens. This crucial event occurs very early in human gestation, specifically during the 3rd and 4th weeks of pregnancy. At this stage, the embryo is incredibly tiny, yet it is already laying the groundwork for its most complex organ system. The hollow neural tube we just discussed will eventually differentiate and grow into the entire central nervous system. The top portion will expand and fold to become the brain, while the rest of the tube will extend to become the spinal cord. Since we now understand how the neural tube is supposed to form, we must ask what happens if something goes wrong. Understanding neurulation is absolutely critical, because if this biological zipper gets stuck and fails to close properly, it results in severe, often life-threatening conditions known as neural tube defects. For instance, if the tube fails to close at the top, near the head, it causes anencephaly, a condition where major parts of the brain simply do not develop. If it fails to close at the bottom, near the tail, it causes spina bifida, which can lead to lifelong paralysis and severe nerve damage. Knowing the severe consequences of these defects, you might wonder how we can prevent them. This is exactly why doctors strongly recommend that pregnant mothers take folic acid, also known as vitamin B9. Folic acid provides the essential cellular building blocks needed to ensure this biological zipper closes smoothly and completely. Without enough of this vital nutrient, the cells cannot divide and move fast enough to seal the tube before the developmental window closes. Proper maternal nutrition is the primary defense against neural tube defects, ensuring the embryo has everything it needs for healthy brain and spinal cord formation. Now that we understand the biology and importance of neurulation, let us look at how scientists study it mathematically. While neurulation is a biological event, biophysicists use mathematical models to understand the speed of this folding process. Just like a physical zipper, the neural tube closes at a specific, measurable speed. We can represent this zipping process using a simple linear kinematic equation. L of t represents the total length of the closed neural tube at any given time, measured in millimeters. L0 stands for the initial closed length. The variable v represents the velocity or rate of closure, telling us how fast the folds are fusing in millimeters per hour. Finally, t is the elapsed time in hours. If velocity increases, the tube closes faster. If it decreases, the closed length will be dangerously short. Before we move on to more complex scenarios, let us see this formula in action with a concrete example of a healthy embryo. Suppose a healthy embryo's neural tube begins closing from an initial closed length of 0 millimeters. The biological zipper moves at a constant, healthy closure velocity of 0.2 millimeters per hour. We want to find out exactly how much of the neural tube has successfully closed after a time period of 24 hours. We substitute our initial length, our velocity, and our time into the equation. Multiplying the velocity by the time gives us the total distance traveled. The final answer is 4.8 millimeters. In physical terms, this means the embryo successfully zipped up 4.8 millimeters of its neural tube in one day, showing steady, healthy progress. So, we have successfully used our formula to find the length. This brings us to an important question. What if we know the required length, but need to find out how long it will take to close? To do this, we must rearrange our kinematic equation to solve for the unknown time variable, t. We start with our standard linear equation. Our first mathematical step is to isolate the term containing time. We do this by subtracting the initial length, L0, from both sides of the equation. This leaves us with the total change in length on the left side, equal to the velocity multiplied by time on the right side. This sets us up perfectly for the final step. Now that we have isolated the velocity and time on one side, let us complete the derivation. To get the time variable completely by itself, we divide both sides of the equation by the velocity, v. By rearranging the formula, we arrive at our final result. This new equation tells us that the time required to close the neural tube is equal to the target length minus the initial length, all divided by the closure velocity. This rearranged formula is incredibly powerful. It allows scientists and doctors to calculate exactly how many hours are required to achieve a specific target length, which is crucial for determining if the tube will close before the biological developmental window shuts. Now that we have derived this time formula, let us apply it to a real-world scenario involving a developmental delay. Imagine an embryo with a slight folic acid deficiency, causing the neural tube to close at a slower rate of 0.12 millimeters per hour. The tube must reach a critical length of 6.0 millimeters to avoid a defect. We plug our target length, initial length, and slow velocity into our new equation. Dividing 6 by 0.12 gives us 50 hours. The biological window strictly closes at 45 hours. Because 50 hours is greater than 45 hours, the tube will not finish closing in time. This mathematical reality perfectly illustrates why maintaining a healthy closure velocity is absolutely vital. Having calculated the severe impact of delayed closure, let us visually examine exactly what is failing to close. Here we see a cross-sectional diagram of the three main stages of neural tube formation. On the far left, in stage 1, you can see the flat neural plate sitting above the notochord. Moving to the middle, in stage 2, the plate begins to dip downward, creating the neural groove, while the edges rise up to form the neural folds. Finally, on the right, in stage 3, those folds have met and fused completely. The top layer becomes the continuous ectoderm, and safely beneath it lies the fully formed hollow neural tube. If the velocity is too slow, the embryo gets stuck in stage 2. We have covered a lot of ground today, so let us summarize the most important ideas. First, neurulation is the magnificent biological folding process that transforms a flat sheet of embryonic cells into the foundational structure of your brain and spinal cord. Second, this delicate zipping process relies on precise mechanical forces and strict biological timing. Failures here lead to severe developmental disorders like spina bifida. Third, proper maternal nutrition, specifically folic acid, is essential to maintain a healthy closure velocity. Finally, we saw how simple kinematic equations allow us to model this process and predict whether the tube will safely close before time runs out. You now have a solid understanding of the incredible, fragile beauty of early neurobiology.